Welcome to another UHS MDCAT video. This time we will be doing the DNA topic from Unit 10 Genetics according to the UHS topic. So first of all, we're going to talk about the four bases that make up nucleic acids. So adenine and guanine are the purines, whereas cytosine, uracil, and thymine are the pyrimidines. You should also know that adenine pairs up with thymine in DNA and with uracil in RNA. And some features that you should remember in case you have to do identification is that adenine has an NH2 group, right, and 2CH and 1 NH, and this is the only thing you need to remember. And then over here with uracil we have 2Cu, and with thymine we also have 2Cu, and obviously the purpose of one of these Cu is to form a hydrogen bond with this NH2, right? So, or to make it easy enough for you to remember, now how uracil and thymine are different, that is in terms of one group. Thymine has an extra functional group, it has a methyl portion to it. Easy enough to remember if you say the DNA being the more prominent, more important one anyways, more prominent would be a better way, or to have an extra methyl group. These points are fairly easy to remember in my thinking, and obviously the ones that will be left will be cytosine and guanine. And the way I remember this is COG. Interesting thing is that this has both a CO portion and an NH2 portion, and cytosine similarly has both a CO portion and an NH2 portion. And if you remember that CG bases form 3 hydrogen bonds, and that is easy enough to remember. If you can see that there are 3 functional groups in cytosine that are capable of making a bond, right, like this, 3 hydrogen bonds, and with guanine we similarly have 3, whereas adenine is capable of making just 2 with uracil and with thymine, and so it just has 2 functional groups capable of making bonds. Same over here, same over here. So on the basis of remembering this from A levels, we already know if you are given this structure and they're asking you what it is, you can easily see it's double ringed, and you're like, okay, this is double ringed, so this has to be one of the purines. It's either adenine or it's guanine. And if it's like 1 single ring, then you're right, okay, you see the cytosine, uracil, or thymine, and you know that cytosine makes 3 of hydrogen bonds with its pair, so it needs to have 3. If it has 3, it's definitely cytosine. If it has 2, it's either uracil or thymine. And then you can look for the CH3 portion to confirm which of it is. So this identification came in 2017, so it's fairly important. You also ought to be able to tell about the deoxyribose and the ribose ring. Both are 5 carbon, 1, 2, 3, 4, 5. And the difference between these is that deoxyribose has 1 less O, and this 1 less O is on carbon number 2. This portion over here is identical. However, over here we have HOH, but we have HH in deoxyribose. This portion is again identical, this portion is again identical, and so is this one. The only difference is at carbon number 2. Otherwise, the two are completely identical 5 carbon monosaccharides. and the ring is 5-sided: 1, 2, 3, 4, 5. But not all of the carbon atoms are part of the ring. If they ask you how many are part of the ring, that would be 4 of them, not all 5, but it's still 5 carbon-sided because of the O sort of like being part of the ring. They might even ask if an oxygen is part of the ring. So if it's O is over here, then obviously it's not over there. Technically it is actually. This one is a mistake. Right. Phosphate portion, we know what is a nucleoside. A nucleoside is basically either deoxyribose or ribose along with the phosphate group. It becomes a nucleotide when nitrogenous base is added to it. Now, phosphate group has this double bond, this OH portion, and these O minuses. And you have to be able to number the carbon atoms. Carbon number 1 is the one to which the base is attached. Can be either one of these bases. Carbon number 2 doesn't have any attachment. Carbon number 3 has the OH portion, and it's this OH portion that sort of like forms a phosphodiester bond with a phosphoric acid from the one below it. And on 5th we have a PO4. Right? Phosphoric acid, this one, PO4, sort of like it can be abbreviated as, right, is on the 5th carbon, the one that is projecting off. The hydroxyl that forms a bond with the one below it is on carbon number 3. The nitrogenous base is on carbon number 1. The thing to remember is that condensation takes place from 5' to 3' direction continuously. This is important because CH2OH, right? So one of the H is given up and NH2 is formed. Because an OH is given from over here and an H is given from over here to form the bond. And the base is over here and this is over here, and this over here is a phosphodiester bond. This is a 5' end because this part is facing 3' end because OH is facing over here. CH2O, PO, another O, and O minus is projecting like this with another C because an H has been given up from this side and an OH from the other side. So one H2 from over here, one H2 from over here to form this single one, and then another one to form. So because it's a diester bond, easy enough to understand, I think. Amount of adenine in DNA is always equal to the amount of thymine, and the amount of guanine is always equal to the amount of cytosine. Equal proportion of the purines and the pyrimidines, if they ask. Right, we have a German chemist and he discovered DNA and he extracted a white substance from the nuclei of human cells and from fish sperm. And he called the substance nuclein because it appeared to be associated with the nucleus. And because it was acidic, it came to be known as nucleic acid. And biochemist P.A. Levine basically figured out the basic structure of nucleic acids, saying that it composed 3 main components: a phosphate group, a 5-carbon sugar, and nitrogen-containing bases called purine and pyrimidines, and that RNA contains uracil instead of T. Concluded that RNA and DNA molecules are made of repeating units called nucleotides, And in a nucleotide, the nitrogen base is attached to carbon 1 of a pentose sugar, and phosphate group is attached to carbon 5 of the sugar. In addition, a free hydroxyl group is attached to the third carbon atom. And the 5' phosphate and 3' hydroxyl groups allow DNA and RNA to form long chains of nucleotides, because these two groups can react chemically with each other. And this reaction is called a dehydration synthesis, because the water molecule is eliminated and a covalent bond is formed that links the two groups, and the linkage is called phosphodiester, where the phosphate group is now linked to two sugars by means of a pair of esters. As you can observe over here, this is one POC and this is another POC. And the 2-unit polymer resulting from this reaction still has a free 5' phosphate group at one end and a free 3 hydroxyl at the other end. In this way, many thousands of nucleotides can pair up. Linear strands, no matter how long, will almost always have a 5' group at one end and 3' free group at the other. And the similar significance of the regularity is pointed out by Jakob, and this is important that this guy was the one who figured out the proportion of the bases when a British chemist, Rosalind Franklin, carried out an X-ray diffraction analysis of DNA. And in this analysis, a molecule is bombarded with a beam of X-rays. When individual rays encounter atom, their path is bent or diffracted, and the diffraction pattern is recorded on a photographic film. When carefully analyzed, this pattern gives a three-dimensional structure of a molecule. And he prepared this in the laboratory of British biochemist Maurice Wilkins, who prepared DNA fibers. The diffraction pattern suggested that the DNA molecule has the shape of a helix, has a diameter of 2 nanometers, so 2 nanometer diameter, and a complete helical turn every 3.4 nanometers. So very important, diameter of DNA molecule is how much is it? It's 2 nanometer, and the length of one turn is 3.4 nanometer. So if they ask you how the helical structure was discovered, it was discovered by Rosalind Franklin with the help of Maurice Wilkins, and they figured out via analysis of a diffraction pattern via X-ray diffraction, the DNA molecule has a shape of a helix. Also the diameter, the approximate diameter, and the helical or the length of each helical turn, both of these points were figured out. This is the 5' end, this is the 3' end, AT, GC, 3 bonds. Now you should take a look at this. So this is a turning right. So this turn over here is short and is called a minor groove, where this turn is long over here and it's called a major groove. Again, this is a major groove, whereas this is a minor groove. A groove is when there is this fissure or constriction. So if they ask you what suggested the double helical shape of DNA, the answer is X-ray diffraction pattern. So these were some important tidbits. So in learning informally of Franklin's results before they were published, two other scientists, James Watson and Francis Crick, they were young researchers at the University of Cambridge. They worked out a likely structure, which we now know to be substantially correct. They proposed the molecule is a simple double helix with the bases of two strands pointing inwards towards each other, forming base pairs as observed. Base pairs always consist of purines which are large pointing towards pyrimidenes, which are small, keeping the diameter of the molecule a constant 2 nanometer. Because hydrogen bonds exist between the bases in a base pair, the double helix is stabilized as a duplex DNA molecule composed of two anti-parallel strands, one running in 3' to 5' and the other in 5' to 3'. The base pairs are planar, obviously, and stack 0.34 nanometer apart as a result of hyperphobic interactions contributing to the overall stability of the molecule. So this distance is 0.34 nanometer and is due to hyperphobic interactions. This is an interesting term. They might ask you whether the interactions are hydrophilic, hydrophobic, or whatever, and the answer is hyperphobic interactions. Between adjacent base pairs, hyperphobic. And these hyperphobic interactions mean that they are stacked 0.34 nanometer apart, and this contributes to the overall stability of DNA duplex. In the double helix, adenine forms 2 hydrogen bonds with thymine, guanine forms 3 hydrogen bonds with cytosine. Adenine will not form proper hydrogen bonds with cytosine even though the CO and the NH2 portions might be present. And guanine will not form hydrogen bonds with thymine. Consequently, if you look at the structure, we have adenine and cytosine. Even these portions are present, proper bonds will not be formed. See, again we have these portions, but proper bonds are unable to be formed, although there will be hydrogen interactions. And the base pairing ensures the equivalent proportionality. This part over here is the sugar phosphate backbone. This is the hydrogen bond. This is the OH 3' end. This is the phosphodiester bond, POC, POC. Hopefully you guys found this to be helpful. I'll make the next video in a while. Thank you.